Howdy, everyone. I'm one of your Blue Jeans and Boots hosts, Anthony Matarazzo, here to let you know that the Interdisciplinary Graduate Program in Genetics and Genomics at Texas A&M University is currently accepting applications for our PhD program. Whether you've listened to our podcast for a while or are new here and have been thinking about going to graduate school, check out our flyer posted on our website at genetics.tamu.edu. Our program is truly interdisciplinary and includes developmental biology, evolutionary genetics, bioinformatics, plant genetics, medical and applied sciences, and so much more. Come learn from researchers in over a dozen departments and find your scientific passion today. The application is free and the stipend is competitive. Learn more on our website at genetics.tamu.edu. Now, let's talk about some science. Howdy, everyone. You're listening to Blue Jeans and Boots, where genetics grad students talk about how to navigate a PhD program while trying to stay sane. We also often invite faculty from our interdisciplinary program here at Texas A&M and pick their brains about their research, what it's like being a principal investigator, and their journey to becoming a professor here at A&M, or in special cases like today's episode, at places other than A&M. In this episode, we'll talk to Dr. George Church. Dr. Church is a Winthrop Professor of Genetics at Harvard Medical School, Professor of Health Sciences and Technology at Harvard University and Massachusetts Institute of Technology, and founding core faculty member of the Wyss Institute for Biologically Inspired Engineering at Harvard University. He received his PhD in biochemistry and molecular biology at Harvard University in 1984, and his research has spanned many disciplines, from genomic sequencing to synthetic biology. He has also co-founded multiple companies such as Colossal Biosciences, which is working to de-extinct various species such as the woolly mammoth. My name is Anthony, and I'll be your host today. Welcome, Dr. Church. Thank you for joining us on the podcast. We are happy to have you here.
It's my pleasure.
So to start off, I wanted to ask what influenced you growing up to pursue a career in science later in life?
Well, I was in a fairly science-free zone growing up in Florida. There weren't even any classes in science until 7th grade, and they had to bring in an extra teacher to do that. And none of my family or friends of the family were scientists. But I did, and I had a slight reading disability. I was dyslexic, so I had a bunch of stuff going against me. But I did like nature, and I would find weird things, like metamorphosis was something I discovered on my own, rediscovered, obviously. And I had to find it in books without being able to read them, so I did it mostly with pictures. So that was really the beginning, and then getting exposed to the World's Fair with all kinds of new glitzy technology, and they're trying to give you a feeling of what the future looked like. And being a 10-year-old, I got confused between the future and what was actually happening, and I got very frustrated. 10-year-old frustrated, and felt like, well, if it weren't really here, I had to somehow help create it.
Right. So what inspired you to pursue a graduate degree? What advantages do you think that having a graduate degree has over just going straight into industry after undergrad?
Well, I'm still not in industry, but as a profession, I think, I wasn't surrounded by industry people. In fact, biotech didn't exist when I was an undergraduate. I think my main motivation, frankly, was I wanted to get paid for going to school rather than paying them. And I knew that was the deal with graduate school. And so I actually rushed through undergraduate as quickly as I could to minimize the amount of cost. And I got a lot of credits during the summer where it wasn't, I was doing summer research, and that didn't cost any money. So I was being very frugal was really the main reason. But I also sort of felt like even as an undergraduate that the real deal was happening in the research labs in the medical school. And so I spent all my time over there taking graduate courses and doing research in the laboratories, because I felt everything I was learning on the main campus was all toy stuff. It was like prefab little experiments. Now, why I didn't go straight into industry, like I said, there was I was getting a degree in chemistry and zoology. I could have gone. There was a chemistry industry that I was vaguely aware of, but I just don't remember any recruiters being on campus. I just didn't wasn't really a known route for me.
Hmm. I mean, the frugal route is definitely something that I think a lot of grad students can relate to, especially these days. Um, but you did end up in the industry realm eventually, right?
Yes.
With Colossal Biosciences, for example. So, could you tell us how the research went from the academic setting and into the industry setting?
Well, way before Colossal. So Colossal started in 2021, and I was already doing industry stuff in 1978. So kind of at the
Mm.
dawn of the biotech revolution, I was there in Cambridge, Massachusetts during the big recombinant DNA brouhaha, and what that debate did was it suddenly got investors excited about biotech. They said there must be something going on here. I mean, they didn't quite know what it was, but it was getting a lot of attention, mostly negative. But that didn't stop the invest. That just made the investors more interested. And so Biogen was formed by my professor. But even before that was really operational, I independently of my professor, uh, I wrote independent of my thesis. I wrote software that was the first software for reading the automated reading of DNA sequence, and somehow Biorad, which is a very different kind of company than Biogen, which didn't really exist yet. Biorad got wind of this, and I would commute to Philadelphia, trying to develop a DNA sequencing instrument, and at the very last minute they changed it into a much lesser instrument. I mean, it's still the same parts. It was a densitometer, and I was a little disappointed that they had taken this beautiful sequencing device and turned it into this thing. It's kind of like into a doorstop. Um, but but in the process, I met a laser physicist named John West, who would later, who didn't know much biology, but we had conversations. I learned some laser, he learned some biology, and he went on to be the guy that ran the first generation um, manufacturing of first generation sequencing devices that sequenced the human genome. And then
Oh
he went
wow.
on to do the second generation, which was Solexa, now called Illumina, which did most of the genome since then. So that was a good investment of our time, I think, even though in the short term, it seemed um, the opposite.
Right, but you were able to use those connections to, help further science and
Yeah,
and collaborate in that sense, and that's
yeah,
awesome.
yeah.
Um, but you did mention that you were up against some kind of skepticism, I guess, as biotech as a whole was, and still kind of is. So how do you combat the people that are still a little wary of genome editing or synthetic biology?
Well, I'm very non-combative, first of all, but I sort of feel like if they don't like it, it means that we're not making the right product,
Mm,
or not communicating adequately. And I think one of the problems with GMOs, which got so recombinant DNA settled down pretty quickly. It was initiated by scientists, and it was kind of solved by the NIH in various lots and lots of meetings, um, and it created, I think, a safer environment for even non-recombinant DNA biology. Even uh, infectious disease was safer, I think, as a result of that discussion. But anyway, fast forward to GMOs. Plants were a very special case, and most of the objection was to plants. So we were making insulin in bacteria, which is a very strange thing to do. But there was no problem with that because it was way better than getting insulin from cadavers. Um, then so, but plants were still moving genes from one plant to another, say, and that was called transgenics. Fast forward to today, and we're making mutations in specific places that aren't moving around big pieces of DNA. And even though that may sound irrational to a scientist, but it's but you have to respect people's opinions about, that to them it sounded kind of creepy to move genes between species. And so the cisgenics is getting a better reception. And part of it is that it's this, you're just changing one base to another. You're changing an ACE to a C, ACGT. And the other thing is that was the politics of a United States monopoly was not popular in Europe, but now fast forward to today, and part of the CRISPR revolution was European via Emmanuel Charpentier, one of the co-inventors, and I think, and there are a lot of small startups that are using new technology like CRISPR in Europe, and so it's no longer an American monopoly. And so I think overall, those two changes gives them a face saving. Also, it's quite evident how powerful it is because at least half of the world is using GMOs and doing well because of it. So I think those three things can combine to greater acceptance. There's always been pretty good acceptance in the United States, except in fancy grocery stores.
Right.
Yeah.
So how do we make the jump from genetically modified crops for food production to bringing back the woolly mammoth?
Right. So another thing that I think has helped the discussion about genetic engineering in the wild is approval of or genetically engineered salmon. That's a semi-wild thing. The crops are semi-wild. They're out farms, but they're mixed in with weeds and the rest. But then climate change is something that the Europeans, who tend to be there's two groups that are most active against GMOs historically, and that was wealthy people in America and Europe. So if you have the luxury that you can have any food you want, then you can say, well, we'll stick to the expensive foods. Uh, so so like a lot of the landed gentry in England are anti-GMO. So, but if you look at the current sets of crops, the current feeling in Europe is very concerned about global warming and green chemistry and so forth, and so where Colossal comes in is we're interested in restoring ecosystems, um, um, restore is sometimes increasing diversity by reaching back into the past and finding things that are missing in current populations that will make them more robust to change, finding things that will allow keystone species to restore an environment. So in this case, the keystone species is an elephant that's adapted to the cold. Elephants love to knock down trees. The trees kind of, there's been a swap between grasslands, which are very fertile and are very good at turning carbon dioxide in the air into fixed carbon in the roots of the grasses, um, much better than trees are. And the trees also, trap snow and has trapped the heat underneath the snow like a down blanket. And anyway, all these things led a bunch of environmental scientists to conclude that it might be better to go back in time to when we had the rich grasslands that were building up big layers of fixed carbon every year, and to keep the frozen tundra frozen, because the, I think what a lot of people don't appreciate is that the topsoil is um, about a meter thick in places that otherwise look very green, like rainforests. But they're constantly turning over. Their wood is rotting and turning back into carbon dioxide, and the topsoil stays thin. While in the Arctic, every year you get a new layer of grass and grass roots and excrement from the animals, and it just builds up over time to when it's 500 meters thick rather than 1 meter thick in the rest of the world. And so there's a lot of carbon. It's a great way to sequester carbon, but it's also fragile in that if that melts, now all the carbon, gets turned over with methane-producing bacteria, and methane is 80 times worse than carbon dioxide, which is a lot worse than frozen tundra. So there's 1,400 gigatons of carbon at risk in the Arctic. Compare that to 10 gigatons of human carbon that we worry about every year. So it's a big deal. So anyway, we're adding our bit. It may not be the biggest way to fight, but it has advantages over all the other ways. It may be the biggest because it's not just slowing down the human component, which is 10 gigatons. It's actually slowing down the kind of almost natural process at this point. It's natural at this point because the humans killed off a lot of herbivores. Anyway, that's the long version. Or the
No
medium long
worries.
version. I have a longer version.
But you touched on a lot of good points. It's really interesting to hear of that plan to combat global warming by bringing back those keystone species. Along with the woolly mammoth, are there any other species that you're looking at resurrecting?
Well, the classic keystone species that was very well studied was the wolf in Yellowstone National Park in the United States.
Right.
So, in that case, it was gone for 70 years. They brought it. They did studies that indicated that the park was in worse shape, um, and they very intentionally got rid of it, and they very intentionally brought it back. And when it came back, it had all the ramifications that they thought. So at first, it scared away the caribou. It didn't kill off the elk and the various large herbivores. It scared them away from the little saplings that they kept, and then that allowed them to become into big trees. The big trees meant that the beavers had something to build dams with, and then suddenly you had a more vibrant aquaculture, um, and all sorts of fish and waterfowl you didn't have. So it suddenly became a much more vibrant environment where everything was in balance. Um, and so we are looking at about a dozen different species that might be like elephants are keystone species in every environment they've been in, whether it's from Africa to Asia to Siberia. So, so usually the top species when they're lost, um, is a big negative, and not just from an ecologist who wants things the way they were, but going forward to the future, I think we want to have a frozen tundra sequestering carbon as fast as possible, so we can get back to pre-industrial levels.
So you mentioned the African and Asian elephants. Colossal Biosciences collaborated with the Vertebrate Genome Project to generate better genome assemblies for these animals. Are there plans to continue collaborations with companies such as VGP, or to continue to generate high quality reference genomes for species?
Yes, so This is something that Colossal can afford that wasn't previously affordable in the conservation community, and so it's a fairly easy gift to give away. Um, and you know, you can do things like manatees and the stellar sea cow, which is like I think 6 times bigger. Um, you can do all sorts of extinct marsupials because of the introduction of non-marsupial mammals into Australia, Tasmania, New Zealand. And there are a lot of species that the indigenous people really love or loved, and they want to see. They're excited about seeing them come back. So I think getting high quality genome sequence is a relatively inexpensive and essential part of bringing back at least some of the traits, many of the key traits of these extinct species or endangered species.
Are there any challenges when compiling these genomes?
Well, there were definitely. Once upon a time, before Svante Pääbo, who got the Nobel Prize for ancient DNA, they were in little tiny pieces. You know, sometimes as 20 base pairs or less, which you can't really do much with. The cytidines, so ACGT, the C's, were deaminated, so they look like U's, which is very different. And so, yes, it was hard, but we built up skills for finding bigger DNA, you know, finding better specimens. Certainly, if you have the luxury of having something that's been frozen since it went extinct, then that's best case scenario. You can get really excellent. But even museum pieces that have been at room temperature the whole time can also be recovered, like the dodo and the Tasmanian tiger was preserved in ethanol, and so anyway, there's the record now is I think 2 million years old is the oldest DNA
Wow,
we can get. Yeah.
that's amazing. So Colossal recently created induced pluripotent stem cells for the woolly mammoth's closest living relative. Why was this such a significant step in the regeneration process?
Well, so we need to do some genome editing to make them cold resistant. Um, we want to be able to check that the edits we've made are not just theoretically the right edits and chemically we did what we said we were going to do. We want to make sure that they're biologically functional. And short of making an elephant, which takes a while, you can test them in small things called organoids that you get out of stem cells. But you have to have stem cells to make the organoids. And the organoids you can test them for, you know, the kind of hair they make or the cold tolerance or various things like that. So that's one reason. The second reason is that we're going to need to make a lot of embryos, which means we're going to need a lot of eggs. And since these are endangered species, you don't want to go in there messing around with their reproductive systems when they're just barely holding it together, making elephants the normal way. So you want them to keep producing baby elephants. So one direction that we're pursuing is making the eggs from stem cells in the lab, which has been done with mice, but so far for no other mammal. We spun off two companies now actually, that are working on applying it to humans for medical purposes, for infertility and so forth. But so we're trying to do it simultaneously for mouse, elephant, and human. So that's another application of the stem cells. It's not absolutely required, probably. So for pigs, for example, we've heavily engineered pigs for transplants into people. 69 edits total so far. And in that case, we did it without stem cells, but it was kind of hard because we do it in skin, or fibroblasts they're called, and they'd senesce. And then before they'd senesce, we'd have to, make an embryo, make a fetus, get the fibroblasts out, and start again. And we don't have that luxury yet for elephants, but we have both options in principle.
Right. So you have a long history of entrepreneurship. Could you tell us a little bit about the organizations you've created and what made you move to start these companies outside of the academic realm?
Right. So it started, as we said, with my consultancy with Biorad, where we almost made a sequencing device, and then with Biogen, I was one of the first Biogen employees when I finished. I wasn't really going into industry. I finished my PhD, and I wanted to hang around for 6 months, um, and so since my boss had gone from being a Harvard professor to being CEO of Biogen,
Mm.
it was relatively easy to get a 6-month job there, and I think I carried my weight while I was there. Um, and then so those were two, but then the first one that I really was involved in starting, it was really restarting a company that had been around since the 60s, arguably the first biotech company, but not the big ones like Amgen and Biogen and Genentech. This was one that was doing small contracts on anti-cancer drugs, and then later it had small contracts to make the first synthetic oligonucleotide reagents, and then the first synthetic oligonucleotides, and then finally, producing proteins in yeast. And in every case, they were a little bit ahead of their time, and by the time the field caught up to them, they had already decided it wasn't profitable, and they had moved on to the next big thing. And so they did expression of proteins in yeast, and then they started mapping the human genome. And at that point, they heard about stuff that I was doing on sequencing, and they said, "Oh, we got to have that." And so I helped them rebrand and reboot as from Collaborative Research Incorporated to uh, uh, Genome Therapeutics Corp. So from CRI to GTC, and we sequenced, and they made a copy of my laboratory. I mean, all the way down to the big roll of paper hanging from the ceiling, and it was a very quirky way of doing sequencing. Wasn't first generation, wasn't second generation, it was kind of one and a half generation, and they sequenced the first genome. The CEO just announced one day, just out of the blue, he just said, um, uh, I'm announcing to the press that we're going to sequence Helicobacter pylori in 5 months. And we all looked at each other and said, what? I mean, no one has ever sequenced a bacterial genome or any kind of genome other than little bitty viral genomes. Uh, and how are we going to even know whether we can do it in 5 months? He says, well, that's what CEOs do, is they make this thing, and we did it in 6, and a lot of the employees didn't see much of their family for 6 months,
Wow.
but they did pull it off. It was the first genome, and they sold it twice. So the business model was you'd sell it non-exclusively, and to Schering Plough and AstraZeneca. Those were the two, for about, I think it was about $75 million, which was a lot of money back in 1994. Um, and they went on. They kept producing genome sequences, and they were at the finish line for the human genome. Sometimes people think, oh, the human genome was industry versus academia, when in fact it was industry versus industry plus academia, where my company GTC was on the academic side, the NIH and then DOE funded side of things. So that was the third experience that I had. Actually, just before that, a company called Millipore, has local Boston uh, roots. We They licensed it, and they started making instruments with it, but they were pursuing too many instruments at once in too many different fields. and they had to drop it. And so as soon as they dropped it, then we relicensed it to the what would become Genotherapeutics. So that was essentially my third and fourth experiences. And then I got involved with a company that's now called Flagship Ventures. Back then it was called Nucogen, and I did my next 7 startups. With them, kind of picking and choosing what they thought was going to be um, of benefit to society and financially. And then we entered a phase where one of my postdocs got into his head that he could start one. He saw me starting, and he said he could start one with my help, and he did a spectacular job. He started it in his bathroom with a little baby that had just arrived. So this is a family of 3 with biotech happening in their bathroom, and he formed a company called Ab Vitro, which was sold to Juno, which then was sold to Celgene. It was a very big transaction. And then he went on to start another company, and then my next postdoc said, "Hey, I can do that. He's not that much better than I am." Um, and then ever since then, pretty much we've led in this new revolution. It's called founder-led companies, so with a big emphasis on even young founders, meaning postdocs, taking the C-level positions, CEO, CSO, and so forth. And I was surprised. They've done very well, straight out of postdoc. We've co-founded. I've been involved in 48 of them so far. There've been a few more that I just kind of helped without being co-founder.
So, what do you think has made you successful at helping mentor these trainees to develop their own companies after they leave your lab?
That's a good question. I think partly we have a culture that's fairly non-judgmental. We kind of the people who try and fail are heroes rather than failures. Um, it's a culture where at NASA and I think justifiably in any life and death situation, failure is not an option. That's the motto. But in a bio research biology environment, we make these things called libraries or mutagenize, make collections of mutagenized organisms. And in there, there's millions of components to the library, millions of different molecules you're testing. And if you fail a million times out of a library that has 1,100,000, you still have 100 successes. And so it's not, the ratio is not important. It's just whether you make it over the threshold and get something out of there. So that's not just a science protocol. That's actually a kind of a philosophy of life too. So I think that's part of it. Or those 2 related things are components. I think also just a lot of it has to do with location. I'm at the intersection of Harvard and MIT, which are 2 pretty decent universities, and we get good graduate students and track pretty good postdocs, and so I'm just kind of coasting on the legacy of the environment, and I think that's true for a lot of university towns. Boston's special in that because it has such an emphasis on biomedical research, like most of the big pharma have at least some kind of outpost here. Then the investors get their mindset, oh, we don't need to get return on investment in 2 years. We have to wait until the clinical trials are over, which takes 10 years. And so they have a longer view. So that helps create the right mindset. And I think the interdisciplinary thing helps. And also it helps that I've been kind of experiencing this since I was, since I was a student starting graduate school. I happened to, have this experience with Biorad, which I'm grateful for, because it got me thinking about how my students would feel when they got the entrepreneurial bug. And I think I was more sympathetic than someone who had been a professor their whole life, and then at age 60 started a company. They might might be less student friendly, let's say. Um, but anyway, the real answer is I don't know.
Well, you're obviously doing something right. I think the having a culture of um, failure is an option in grad school is really helpful for both the mentor and the mentee to manage their expectations and to not get too overwhelmed when things don't go the right way because that is science. So learning to get used to that, sitting in the failure, I know is hard for a lot of try hard grad students who want to be the best and at what they do, and so it's nice to hear that having students that perform at that level still fail and being okay with that, and being able to move forward from that is really good to hear.
Amen.
So you mentioned a little bit about the Human Genome Project. So one question I wanted to ask is, what do you think of the idea of personalized medicine and personalized genomes? Is that a reality? Do we have a long way to go, or is that something that we can expect in the near future?
So I have a kind of a love-hate relationship with personalized medicine and precision medicine. I think I was one of the pioneers in it, and in fact, before the Genome Project was really even over, I started thinking about the Personal Genome Project, which I started in 2005. Um, but the goal there wasn't necessarily, well, anyway, part of the goal was to develop personalized and precision medicine. But part of it was to explore new technologies that you could use in general. It was to be able to share information and share cell lines that are properly consented. And as a consequence, we became the standard for the National Institute of Standard and Technology. So all of our genomes are the standard genomes. But so in the Personal Genome Project was heavily committed to precision and personalized medicine. And we had a well-consented cohort. But there's another part of my lab and uh, where we're working on more generic things, in particular, very common diseases, and the most common of all is aging. So you asked whether personal and precision medicine is a thing already, and it is. I think cancers are increasingly stratified by the kind of cancer and the genes that are involved, and that determines your treatment. There certainly has always been personalization of diseases that were diagnosed on birth. So almost not everybody knows this, but almost every baby born in hospitals for decades now have been tested for 40 genetic functions, and they're things you can. They're all actionable. They're things you can do. You can change the diet or so forth. Um, so, so precision medicine is high and personalized, is alive and well. But I think the big breakthrough is going to come in more generic medicine because it's going to, first of all, it's going to be affordable. And when I put on my bioethics hat, it's not just safety and efficacy; it's also affordability. We've helped bring down the price of reading genomes by 20 million fold to make it affordable for everybody. We have to do the same thing for therapies, and the personalized therapies, the gene therapies for rare diseases, are going through the roof. They've gone up eightfold over the last 8 years, and they're now around $4 million a dose,
Wow.
because the R&D is the same as any other drug, but the number of people using it is in the hundreds, and so the denominator is so small that you have to. And the Orphan Drug Act in the United States and makes that possible, profitable even, because before people just weren't taking any chances on rare diseases, but now they are. But now we've kind of overcompensated. There's um, a lot of emphasis on rare diseases, but anyway, there's plenty of opportunities and breakthroughs in medicine that is making age-related diseases suddenly become very feasible to do, and even with gene therapies. So gene therapies can be less than $100, even though the rare ones are 4 million. If you got something that affects, say, 6 billion people out of the 8.2 billion on Earth, which is the the COVID-19 vaccines were formulated as gene therapy. So they either had viral capsids or lipid nanoparticles with single genes inside. So that's exactly how all the other gene therapies are made. And those were on the order of 20 some dollars, um, without considering government support. Just that's the raw cost. So that'll probably creep up to $100 at some. But the point is, it's affordable, and in a public health sense, the governments definitely should want to do that kind of gene therapy rather than paying the incredible costs of that happen at end of life, typically. So it's happening. The precision medicine, personalized medicine is happening, but it's not the only game in town.
Right. And it seems like that targeting specific diseases that don't affect large populations of people is happening, but not at an affordable rate.
Right, and what sometimes is neglected in the conversations is that there is a very affordable alternative. So
Mm,
whenever you're developing a technology, it's not, you have to think about what the competition is, and it's not just a price thing. It's quality of life and so forth. And so the alternative is genetic counseling, or if you do genetic counseling late, then the person you fell in love with is incompatible in a certain sense. You're going to have a high fraction of your children will be severely um, damaged, diseased, and so that means that one or both parents will have to quit their job and be full-time caretakers and fundraisers um, for charitable organizations. So if uh, if you do it late, like during pregnancy, then your choice is termination, which is not acceptable to a lot of people in the world. If you do it earlier, um, um, then you're either going to be adopting or something like that. If you do it even earlier, then you're going to be deciding who you're marrying after having fallen in love. That's not good. If you go even earlier still to the matchmaking, not dating, but matchmaking, where you're really aiming to have children, then you're only introduced to people who are genetically and maybe in other ways, the way that matchmakers take all sorts of things into account when they say you should meet so and so. And the best ones are getting compensated incredible amount. I mean, it's much more expensive than dating, but it's much more rewarding. So anyway, that if you do it there, then there's no real medical consequences to false positives, which is when you say that two people are incompatible when they're not, because it's only 3% that are going to be incompatible genetically, and it's not incompatible. It's just you're avoiding a series of very serious medical decisions. 3%, and if you don't meet 3% of the possible people you could date or marry, it doesn't make any difference. Typically you're only going to marry one or two, and um, and you won't notice that three aren't on the list. So that's the ideal time to do it, but that hasn't emerged much, except Doria Sharim does do something kind of like that, more at the dating stage rather than the matchmaking, or sorry, at the premarital stage rather than the matchmaking. Um, but it works terrifically well in that community. They've reduced that kind of disease by about tenfold, almost eliminated it.
So Across multiple decades of research and innovation, what do you hope that your legacy is? What do you hope to leave behind for the next generation of scientists?
Yeah, it's a good question. Well, one thing is I am going to leave behind the next generation. That is to say, the legacy are the scientists themselves. I'm very proud of. We have about 300 alumni,
Wow.
and they actually uh, came back, and 200 of them came back a decade ago, and kind of had a reunion. And now I hear about little reunions wherever they go to meetings. There's a bunch of them. Very often, they're these founders of these companies, and they like each other. They're not like the shark the sharks that you see in Hollywood movies, and actually you see in real life, unfortunately too. Um, they're very sharing and supportive of their generation and subsequent ones. So um, I think that's a big legacy. I should also be somewhat proud of our little role in next gen sequencing, in next gen editing or CRISPR, um, alternatives to CRISPR, applying those editing methods to pigs and elephants for transplants and environment. But it's mostly the people. I mean, it's what it's fun day to day to interact with them. Whether they're going up or whether their experiments are going well or not. It's still pleasure to work with these very mature students. You know, um, yeah, that's probably the legacy.
Yeah, that's great to hear. So do you have any advice for current graduate students or those thinking about pursuing a graduate degree?
Advice for them. Well, it's you know everything I'm saying is biased, of course. That's maybe the point. But I would say now is a terrific time to be in synthetic biology. And it's also and I think that there's a lot of enthusiasm about artificial intelligence. I think a lot of what artificial intelligence is producing right now is just art, not just art. I mean, art is important, but it's a it's 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 kind of cool either because they're doing something that humans don't do well, like let's say adding multiplying big numbers or doing a game of Go or chess or Jeopardy, um, which involve a lot of memory and computing, um, or they're creating art where if they make something that sounds like human speech, then you say, oh, that's really they write text really fast. That's kind of cool, but very often it's inaccurate or it's not achieving the goal that you want. But we kind of that's okay, and if and it's and it's okay that they are using 200 kilowatts of power while the human brain is only using 20 watts of power. We kind of say, oh, that's okay. They're still pretty fast. We'll give them the extra juice. Um, anyway, I think that one of the real things that AI is delivering on it's not chat. It's not. It's more. Applied to synthetic biology in protein design, for example, is a big deal. 5 of my companies since 2019 have really leveraged AI plus protein design. So that so I would say that those two fields are looking very promising in the intersection of the two, and it may even be that biotechnology is capable of producing a computing resource that's better than current computers that are more hybrid bio because it still is the case, even though they can do Jeopardy very well. I haven't seen a AI system that's been able to outdo Einstein in 1905. Um, so, I mean, it's one thing to imitate, um, you know, a, a Friday night beer chat. It's another thing, where anything goes. But hybrid bio and silicon might be an interesting future.
Definitely, definitely. So the last question I wanted to ask was, do you have any hobbies or things that you do to stay sane? I'm sure your life is pretty hectic. So anything that you'd like to share?
Um, yeah, I try to keep it low in the hectic department. Part of it is just being very calm. I'm kind of maybe pathologically calm. Uh, but the other thing is, you know, when my daughter was growing up, whatever hobby she had would be my hobby, and I'd learn all kinds of new things. Like she was, uh, you know, big on fashion design. She was a good fashion designer, so I learned all about that. And now her her daughters, um, I watch I enjoy watching their hobbies. They become mine. So um, I had a lot of very risky hobbies when I was young, which I've given up because I sort of, like vertical rock climbing and, you know, down downhill biking. Um, but uh, but I've replaced them with all these other things, and it's fine. I sort of feel like society has spent a little bit too much on my education for me to just flush it down a ravine or a
That's very fair. Well, having family is a great hobby to have. Definitely a good way to stay sane. Have a good support system.
yeah. It certainly gives you balance. It puts things in perspective. It doesn't necessarily make you more sane. I mean, hopefully, it doesn't make you insane either. It's just it's just really gives you perspective. That's the key thing. Yeah.
For sure. With that, I'd like to thank you for joining us on the Blue Jeans and Boots podcast, Dr. Church. It's been a pleasure having you,
Been a pleasure being here. Thank you for great questions.
of course. So today's episode was hosted by Anthony Matarazzo and produced by Isabella Childers. This podcast is brought to you by the Texas A&M Interdisciplinary Graduate Program in Genetics and Genomics. If you have any questions about our program here at A&M, please feel free to email us at bluejeansandboots@gmail.com. You can also find us on Facebook, Instagram, and X at Genetics Podcasts. Thank you for listening, and we hope you tune in next time.